In this video, we will be discussing point mutations seen in DNA sequencing and transcription and translation. Now we are going to talk about the types of point mutations as well as transitions and transversions. Firstly, a point mutation is a mutation in the DNA or mRNA sequence that only impacts a single nucleotide. Point mutations also involve the substitution of a base, being adenine, thymine, or uracil, cytosine, and guanine with another base, which can be classified as a transition or a transversion. A transition mutation is purine to purine or pyrimidine to pyrimidine conversion, while a transversion mutation is purine to pyrimidine or vice versa. Now on to the types of mutations that can occur within point mutations, which are missense, nonsense, silent, and read-through mutations. Missense mutations are when a transition or transversion results in coding for a different amino acid. Nonsense mutations are when a transition or transversion results in a stop codon, cutting the amino acid short. Silent mutations are when a transition or transversion results in coding for the same amino acid. Lastly, read-through mutations are when a transition or transversion results in converting a stop codon to another amino acid, extending the amino acid chain and the reading frame to the next available stop codon. So now we will look at the following DNA sequences and determine which has a 4 amino acid sequence in vivo. So we have our top and bottom strand, and we're going to determine which strand is our coding strand. We find that the bottom strand is our coding strand, and we will flip its direction so we look at it 5 to 3' for the rest of the video. After this, we will change the thymine base pair to uracil to make it the mRNA strand sequence. Now we will find the start codon, which is AUG in the strand, and we will read the subsequent letters in our reading frame until we get to the stop codon. After reading, we find that we have methionine, valine, serine, lysine, and then our stop codon, giving us a 4 amino acid strand. Now we will look at the different possible point mutations. If we look at our original strand, it reads for methionine, valine, serine, lysine, and a stop codon. If we are looking at the next strand on the slide, we have changed a uracil to a cytosine, which will be classified as a transition, and it leads to the mRNA coding for a different amino acid, resulting in the strand coding methionine, alanine, serine, and lysine, which would be a missense point mutation. On the next strand, we changed an adenine to a uracil, which is a transversion, and this results in a stop codon being produced, which is a nonsense mutation. On the third strand, we see a cytosine change to a guanine, which is a transversion that still codes for valine and leaves the strand the same as the original with a slight change in its sequence, which is a silent point mutation. Lastly, we see an adenine change to a uracil, which is transversion, and this results in our stop codon now coding for tyrosine, extending the reading frame on the strand, which is known as a read-through mutation. And here are my references.